This is the second video on photosynthesis. In this video, we'll be discussing the first stage of photosynthesis, the light reactions, where energy from sunlight is transformed into chemical energy. Light is a type of kinetic energy called electromagnetic radiation. Packets of light called photons travel with a characteristic wavelength and energy. Visible light is only one portion of the electromagnetic spectrum, the part that our eyes are able to detect, and includes the range of wavelengths used in photosynthesis. The wavelengths of visible light are from 400 nanometers to 700 nanometers. A wavelength of light is the distance from the peak of one wave to the peak of the next. For example, in purple light, the peak of one wave to the peak of the next would be 400 nanometers. While in red light, the peak of one wave to the next would be 700 nanometers. You can see that the peaks are closer together in the purple light than they are in the red light. The shorter the wavelength, the more energy that photon of light contains. This can be observed by looking at the classification of light waves on either end of the visible light spectrum. Examples of light waves shorter than 400 nanometers are things such as UV light and X-rays, which are known to damage cells and tissues, while light waves bigger than 700 nanometers include things such as microwaves and radio waves, which are not harmful. In photosynthesis, pigment molecules in the chloroplast are able to absorb some of the visible wavelengths of light, capturing the energy in those photons. When wavelengths of light are absorbed, they are no longer able to be detected by our eye. When you observe color, what you're observing is the wavelengths of light that are being reflected from a substance and picked up by your eye. This means that when you look at green leaves, it is because the wavelength of light not being absorbed is in the 495 to 500 nanometer range. This wavelength of light being reflected is why leaves look green to our eyes, because that wavelength of light falls here in the green range of visible light. The main photosynthetic pigment in chloroplasts is chlorophyll, and the wavelengths of light that various chlorophyll molecules are absorbed are shown in this green line. Chlorophyll and other accessory pigments are located in complexes called photosystems, represented by this purple structure. These photosystems are the functional and structural units that absorb light energy. They are found in the thylakoid membrane of the chloroplast. Each green circle represents an individual pigment molecule. Photon energy absorbed by one pigment and then is passed from pigment to pigment until it reaches a specifically configured pair of chlorophyll molecules known as the reaction center. Chlorophyll A in the reaction center will then pass that energy to an electron, increasing its energy level. We describe this as exciting the electron. This absorbed energy will allow the electron to be transferred from the photosystem to another molecule, causing the photosystem to become oxidized. There are two different photosystems in the thylakoid membranes, named photosystem 1 and photosystem 2. Unfortunately, the photosystems are named in the order they were discovered and not the order in which they function. This means that photosystem 2 comes before photosystem 1 in the overall mechanism of the light reaction. An electron transport chain connects the two photosystems together. Once the reaction center of photosystem 2 has absorbed light energy and transferred it to the electron is passed to the first complex of the electron transport chain. When the electron gets to the end of the chain, it is transferred to the reaction center of photosystem 1, reducing it. Once the reaction center from photosystem 2 loses the electron, it is no longer able to absorb light. This means that the electron must be replaced in order for the process to continue. After absorption of light energy, electrons are passed from photosystem 2 to a molecule called plastoquinone, which carries them to transfer to the cytochrome B6F complex. The complex then passes them to plastocyanin to carry them to photosystem 1. During this process, cytochrome B6F functions similarly to electron transport chain complexes of cellular respiration. It uses the energy from the excited electron to transport hydrogen ions from the stroma of the chloroplast into the lumen of the thylakoid, creating a higher concentration inside the thylakoid than in the stroma. This proton gradient can then be used to flow back through ATP synthase, generating ATP through oxidative phosphorylation in the same mechanism as was observed in cellular respiration. It is important to note that this ATP is not used to power all the plant's metabolic processes. Plants have mitochondria to do that. This ATP is specifically made in the stroma of the chloroplast to power the second stage of photosynthesis, which is called the Calvin cycle. This cycle will be discussed in another video. At this stage of the light reaction, we have excited an electron and then moved it from photosystem 2, reducing it, to photosystem 1, oxidizing it. Along the way, the electron has lost its absorbed energy because the energy was used to transfer hydrogen ions. Two things must now happen to keep the light reaction going. The first is that we must replace the electron that has left photosystem 2. In other words, we need an electron donor. The electron donor in photosynthesis is water. Water molecules are oxidized, providing the electron for photosystem 2 and producing oxygen gas and some leftover protons in the process. The protons help contribute to the overall concentration gradient forming in the thylakoid lumen. Now that photosystem 2 has another electron, it can again absorb light energy, exciting the electron and transferring it down the electron transport chain. However, since photosystem 1 still contains the original electron, the electron transport chain will back up and soon stop running. We need an electron acceptor to continue to provide electrons for the light reaction. We also need an electron donor to remove electrons from photosystem 1. However, the problem is the electron that was transferred has lost all of its energy along the way. It must be excited again before it can be removed from photosystem 1. Another photon of light is absorbed by this photosystem, which excites the electron, allowing it to be transferred to ferredoxin, and then to the enzyme NADP+ reductase. The enzyme transfers the electron to the electron carrier NADP+, reducing it to NADPH. Now that the electron has been removed, the chain will be able to continue. The NADPH produced is also required by the Calvin cycle, along with the ATP. The Calvin cycle requires 2 molecules of NADPH and 3 molecules of ATP for each carbon dioxide that it will process. Each molecule of NADPH is carrying 2 electrons. So to make 2 NADPH, 4 electrons must be transported through the photosynthetic electron transport chain. However, the transport of 4 electrons does not produce enough of a proton gradient to produce the 3 required ATPs. Because of this, there is a second pathway for electrons to travel called the cyclic electron transport. In cyclic electron transport, when electrons reach the ferredoxin, here, ferredoxin can instead shuttle them back to plastoquinone instead of on to NADP reductase. As the electrons from ferredoxin are picked up by plastoquinone and transferred back to cytochrome BCF, additional protons are transported from the stroma to the lumen, increasing the gradient that can be used to drive the synthesis of ATP, so that enough energy is produced to power the Calvin cycle.